So just 1 second. Let me change the video settings. Okay, let's start the continuation of all this thing. Okay, so for what we discussed is about your carbohydrate. So followed by the carbohydrates, we'll have to discuss our what is that? Yes, of course, we'll have to discuss our what is that? That is your carbohydrates as done. Amino acids should be discussed. Okay. So let's talk about the amino acids. So I'm just waiting for 1 minute for everyone to join back once again. So come on, guys, join back quickly. We'll start off. All right. So whenever we are speaking of carbohydrate, especially glycogen storage disorders, we read the question very carefully because as I have set the trap right now, okay, so it will it will confuse you people. Sir, can you explain gluconeogenesis and Fenton's reaction before moving forward? Sure, my dear. So somebody asked about this one. Let me explain the gluconeogenesis. What is gluconeogenesis? Very simple, sir. Gluconeogenesis. Gluconeogenesis. First, let's understand the definition of the gluconeogenesis. Gluconeogenesis. So gluconeogenesis is nothing but formation of which substance? Formation of glucose. Of which one? Of glucose from where? From non-carbohydrate source. Okay, so normally glucose is a carbohydrate. So we are using some non-carbohydrate substances and forming the glucose. Okay, so non-carbohydrate source. Okay, so it is from the non-carbohydrate source. Okay, so if it is from the non-carbohydrate source, what is the question we are going to encounter, my dear student? What exactly is the thing we need to understand? So basically we have some one amino acid called as alanine. We have an amino acid called as alanine. We have an amino acid called as alanine. So alanine in the muscles, what will happen in the muscles? Alanine combines with the ammonia, NH3. Okay. So what is this ammonia comes from? So all the amino acids break down and form the waste product called as alanine. Alanine is combined with ammonia. First, before even going there, so we have one amino acid alanine formation that occurs from your glutamate. This glutamate combines with the ammonia to form what is that? In the muscle, alanine will form. Okay. So alanine will form. How it will form? Glutamate takes up all the ammonia. It will take up one more substance called as pyruvate. Pyruvate is a product which is formed as a result of what? Pyruvate is a product which is formed as a result of your glycolysis. So glycolysis hua tha, glycolysis has happened. So after glycolysis, what is there? That pyruvate combined with your what is that? That combined with your glutamate to form the alanine. Now this alanine, my dear students, via blood, it enters the blood. From the blood, it will reach to the liver, sir. Via blood, alanine came to where? Into the liver. Alanine came into the liver. So alanine in the liver will give two substances that is only called as your glutamate. And one more is, it is a combination of pyruvate and glutamate, right? So it will form what is that? Pyruvate. Now this pyruvate, what will happen? This pyruvate, do you remember conversion of glucose to pyruvate is glycolysis. All the reactions which has taken place in where? Yes, in the glycolysis will become reverse. So basically pyruvate will go back and form the glucose using some ATPs. Usually it uses how many ATP? 6 ATP are utilized. From the 6 ATP, it will form the glucose. So this formation of the glucose from the carbohydrate, this reverse only called as glucose alanine and a Cori cycle. That is only your gluconeogenesis. Okay. That is only your gluconeogenesis because we formed the glucose, but we used what? Pyruvate for it. Pyruvate is not a carbohydrate, but without using a carbohydrate, we created a carbohydrate called as gluconeogenesis. Okay. Then one more what you asked was your Fenton's reaction, right? Fenton's reaction. Okay, let me teach you people about that Fenton's reaction also. Now Fenton's reaction, my dear students. So let's talk about where do we take, let's take another page or somewhere we'll explain. So, okay, Fenton's reaction actually is a very important, very easy to understand. In the white blood cell, my dear students, we will have a cell that is your white blood cell. Neutrophil will be there. Into this neutrophil, we have an enzyme called as NADPH NADPH oxidase. NADPH oxidase. So this NADPH oxidase enzyme utilizes the NADPH and sends the whatever the oxygen is there, that will send it in the form of something called as O2 with a minus. We call it as a superoxide. Superoxide. Okay. Now this superoxide, my dear students, enters into something called as MPO halide system. MPO halide system. MPO halide system. Okay. And one more reaction called as Fenton's reaction. Now, what is this Fenton's reaction? Fenton's reaction is a reaction in which what will be there is formation of the free radicals will occur. Formation of the free radicals will occur. So, because of this Fenton's reaction and MPO halide system, we get something called as OCl minus called as hypochlorite. So, basically, what is Fenton's reaction? It is a reaction which is used in the WBC for the formation of free radicals such as OCl minus, which will go and kill the microorganism. So, for the bactericidal effect will be achieved with the help of OCl minus. Hope that part is clear for everybody. Clear? I don't know your name. It's just written iPad A. That's clear for you? Okay. Let's continue. Next, let's talk about the amino acid. So, I'll just quickly discuss the classification of amino acid. Classification of amino acid. So, amino acid without amino acid. Whenever we are speaking of amino acid, let's talk about the little basic things. Amino acid consists of two functional groups. One is, of course, your ammonia, and one more is COOH. COOH is the acid. COOH is the acid and NH3 is the base. That is amino group. So that is why we have a name called as amino acid. All the amino acid combines together to form what is that? Proteins. All the amino acid combine together in a different sequences to form a different types of proteins. Okay. So now, whenever we are talking about structure of the amino acid is an important point to be remembered. Structure of the amino acid, we will have a R. R is nothing but what is that? R is nothing but short form for the long chain of carbons. Short form for the long chain of carbon atom. So it will have one carbon which is bound to COOH group and also it is bound to ammonia group. Okay. So in the throughout the chain, we need to have two functional groups to call it as amino. COOH group and NH3. COOH group and NH3. So R and COOH. Okay. So these two groups are present. So if more number of COOH is there, acidic amino acid. More number of COOH is there, acidic amino. More number of ammonia groups is there, we will call it as a basic amino acid. So we have two types of amino acid, acidic and basic amino acid. So acidic amino acid has more acid groups, that is your COOH or carboxyl group, and ammonia groups if they are more in name number. So for example, in a amino acid, we found an amino acid with 3 NH3, 3 ammonia groups. Is it an acidic amino acid or a basic amino acid? 3 NH3 groups and 1 COOH group. Now you tell me what is it? Is it a acidic or basic amino acid? In one amino acid, we had 3 ammonia groups and 1 COOH group. What type of amino acid? Is it acidic or basic? Everybody should tell me. The answer is more number are there. Very good. Everybody, people are correct. It is a basic amino acid. Basic amino acid. Okay. So once that is clear for you people, let's understand, dear students, what is that basic amino acid? Very good. Very good. So we have something called chiral carbon. With what is that? Chiral carbon. What is that chiral carbon? Chiral carbon is nothing but a carbon atom. There is one carbon atom in the amino acid. It is bound to one side, it was bound to NH3, one side it was bound to COOH, one side it was bound to hydrogen, one side, for example, it is bound to, let's imagine some OH group. Okay, it is bound to the 4 different groups. Any carbon atom which is binding to the 4 different groups, we will call it as a chiral carbon. We'll call it as a chiral carbon. So any amino acid can have a chiral carbon, except amino acid with no chiral carbon is glycine. That is why it is called as simplest amino acid. It is a simplest amino acid. Okay, all right. So we have amino acid. Amino acid means what, sir? We have ammonia group which is there. This ammonia group, for example, it is bound to some ring here. Let's imagine it is bound to a benzene ring. Let's imagine it is bound to a benzene ring. Now, ammonia group, is it free or it is combined to something? It is combined to something. If ammonia group is not free, that type of amino acid, we will call them as a amino acid. The example with, if a amino group is bound to ring or a amino group is not free, we will call it as amino acid. The classic example for the amino acid we have is proline. But what is that? Proline, sir. What is that? Proline. Okay. So that is the story of your proline. Okay. Now let's continue to the next one. One amino acid binds with another amino acid. How? One amino acid binds with another amino acid with the help of a peptide bond. With the help of a peptide bond. So peptide bond is present between what and what, sir? So let me explain you people. One amino acid is there. Let's take one more amino acid. So COOH and NH3. So both are amino acid? Yes, sir. Both are amino acid. The peptide bond forms where? The peptide bond formation occurs between the amino group of one amino acid and COOH group of another amino acid. One amino group of one amino acid binding with COOH group of another amino acid. We will call it as a peptide bond. So they are formed with a peptide bond, and here is the place. One amino acid COOH binding with the another amino acids amino group. Which group? Amino group. We will call it as what? Yes, we'll call it as a peptide bond. Okay. So amino acid from one carboxyl group of one amino acid and amino group of another amino acid. That's what I explained already. Now, classification of amino acids. So, simple amino acids, we have two amino acids which you need to remember, called as glycine and alanine. Repeat, which are the simple amino acids? Repeat with me. Simple amino acids are glycine and alanine. Which are those? Glycine and alanine. Glycine and alanine. Then we have branched chain amino acid, which can be remembered with a mnemonic. Repeat with me. L for leucine, I for isoleucine, V for valine. So what are the three things you need to remember? Leucine, isoleucine and valine. Leucine, isoleucine, valine. So these are the branched chain amino acids. Okay. Then we have sulfur containing amino acid. We have methionine and cysteine. We have what is that? Methionine and cysteine. Then we have hydroxyl containing amino acid, that is your serine and threonine. Hydroxyproline. So these are the types. Okay. Now we have something called as derived amino acid concept. What is this derived amino acid? Now all of you remember, in a protein, all amino acids are genetically coded. Means which amino acid should be there, that information is genetically coded in the DNA. Okay. So if I have a protein with 20 amino acid, the sequence will be there. Now, which amino acid is where? That sequence is coded in the form of a genetic code. If it is coded in the form of a genetic code, so now what I'm trying to tell you is that if amino acid is not coded by any codon, then we will call it as derived amino acid. What is that? Derived amino acid. So classically, we have one amino acid called as hydroxyproline. Okay. Then we have, normally we have 20 amino acid. Later we found out to have two more. One of them is 21st found amino acid is selenocysteine. After later we found out one is called as pyrrolysine. So which is the 21st amino acid? Selenocysteine. Repeat everybody. Selenocysteine. Repeat. Selenocysteine. 22nd amino acid is called as pyrrolysine. Okay. Selenocysteine is coded by UGA. Pyrrolysine is coded by UAG. All right. So once we understood about that, let's talk about the amino acid or proteins. How exactly the fate of the proteins, how your body handles whenever you eat any protein, how exactly your body handles. Let's learn about that. Okay. First, from the food consists of proteins. Okay. So they will come into the GIT. So all the proteins are made out of what? Proteins came into the GIT. All the proteins are made out of what? Yes, come on, tell me. All the proteins are made out of amino acid. Very good, sir. Amino acid. So these proteins will break down themselves into amino acids. A stands for amino acids. Now these amino acids are absorbed. Okay. Absorption will take. After the absorption, these amino acids go into the body. Inside the body, they form the protein. They form the proteins. Form the proteins means you are not using the protein directly, which is taken in the diet. Your body will synthesize the new protein using those amino acids found in these proteins, whatever you ate from the diet. Now, the proteins, whatever formed in the body, proteins do their action, whatever the action may be, enzymatic action or structural action, whatever the action they will do. They will do their action. Followed by the action, proteins break themselves into amino acids. Proteins break themselves into proteins. Amino acids formed from the proteins. Amino acids are formed from where? Formed from proteins. Formed from proteins. Okay, amino acids formed from the proteins, whatever is there, these amino acids will further undergo the degradation. So once amino acid undergo the degradation, they give rise to a toxic product called as NH3, that is also called as ammonia. So ammonia is the end product of your amino acid. Ammonia is the end product. So this ammonia, which is the end product, which is very toxic to the body, how your body handles that ammonia, let's talk about it. Okay, so to talk about it, we need to learn about the urea cycle. Okay, what is that? Urea cycle. So when I'm speaking of urea cycle, first and most importantly, let's understand in the brain, sir. In the brain, whatever the ammonia is present, that ammonia will be taken by glutamate. Okay, and this glutamate, after taking the glutamate with ammonia, we will call it as glutamine. We'll call it as a glutamine. Now this glutamine is transported to the liver. Glutamine comes to the liver. So they will ask you which amino acid transports ammonia. The waste is formed. The waste ammonia is formed in the brain. That waste ammonia, whatever is formed, that will be taken by one amino acid called as glutamate, and that will be transported in the form called as glutamine. Okay, glutamine comes to the liver. Now, one more place in the muscles. Is in the muscle. Okay, so in the muscle, what will happen in the muscle, actually we have glutamate only. In the muscle also, first we have glutamate only. Now this glutamate combines along with pyruvate, forms the alanine. Now this alanine will go to the liver. So which amino acid carrying ammonia? In the form of alanine. So from the muscle, ammonia is transported by your which amino acid? Alanine. And from the brain, which amino acid is transported? Glutamine or glutamate. So you should not get confused. The transporters of ammonia are two, glutamate and alanine. Glutamate from the brain and from all the skeletal muscles of your body, who is carrying alanine. Okay, so they take the ammonia, give it to where? To the liver. So all the ammonia comes into the liver. Once ammonia comes into the liver, what will happen? Let's see. Okay, once the ammonia comes into the liver, what will happen? Let's understand. Okay, so once the ammonia comes into the liver, ammonia combines with your carbon dioxide. Where exactly that occurs? In the mitochondria of the liver. So in the mitochondria, ammonia combines with your, what is that? Yes, sir, ammonia combines with your carbon dioxide with the help of an enzyme called as carbomyl phosphate. The name itself is very clear. It is having two names, carbomyl. Carbo and amine, carboamyl. Okay, so with the help of an enzyme called as carbomyl phosphate synthase 1, it will forms the carbomyl phosphate. Now this carbamyl phosphate combines with ornithine with the help of an enzyme called as ornithine transcarbamylase, and now it will form a citrulline. But citrulline is formed where, sir? In the cytoplasm. Do not get confused. Citrulline formation occurs in the cytoplasm. This citrulline, under the influence of an enzyme called as arginosuccinate synthase, it will create arginosuccinate. Now this arginosuccinate, my dear students, will get converted into arginine. Now arginine, under the influence of one enzyme called as arginase, it will give rise to ornithine and our waste product, that is our urea. Our waste product, that is our urea. That urea will be removed by your kidney. So ammonia is getting converted into urea with the help of a cycle called as urea cycle, which takes place where? In the liver. Which takes place in the liver. In the liver, where exactly? Only in the cytoplasm, only in the mitochondria. It takes place both in the cytoplasm and mitochondria, because it starts in the mitochondria, ends in the cytoplasm. And the amino acid which is forming the urea is arginine. Amino acid which is forming the urea is arginine. Do not get confused by aspartate. Aspartate might play a role, but which amino acid forms the urea? Urea is formed from arginine. Urea is formed from arginine. Okay, so that is an important point to be understood. Okay, so that is your, what is that, urea cycle. Okay, so let's talk about the important aspect, the rate limiting enzyme of the urea cycle. Rate limiting enzyme is carbonyl phosphate synthesis step. That is only carbonyl phosphate synthase. The name of the enzyme is carbonyl phosphate synthase 1. Very good, bacho. What is that? CPS1. Okay. Which amino acid plays a role? That is your arginine. Okay, so there is a disorders of urea cycle. Disorders of urea cycle. What is the disorders of urea cycle? Disorders of urea cycle are due to, what is that, deficiency of which enzyme? Carbonyl phosphate synthase 1, CPS1 deficiency leads to hyperammonemia type 1. Which is that? Hyperammonemia. Hyperammonemia type 1 is due to deficiency of which enzyme? CPS1. Type 2 hyperammonemia is due to deficiency of which enzyme? Ornithyl transcarbamylase. So, if there is a deficiency of CPS1, there is an increase of ammonia. There is an increase of ammonia. If there is a deficiency of OTC, let's see. So, if the ornithine transcarbamylase, if this enzyme is not there, both carbamyl phosphate will increase and ammonia will also increase. Both of them will increase. So, there is an increase of carbamyl phosphate, okay, carbamyl phosphate, and also increase of NH3, that is ammonia. Okay, so that is your ammonia. Both of them will be elevated. That is only your type 2 hyperammonemia. Okay, now if there is a defect of urea cycle, if there is a defect of urea cycle, okay, if there is a defect of urea cycle, automatically ammonia will not get converted into urea. So there is an increase of ammonia in the body. So increase of ammonia is very toxic to which organ of your body? Ammonia is toxic to one organ. Which is that organ? Come on, quickly tell me which organ it is. It is very toxic to which organ? Everybody should tell me. Ammonia is toxic to, come on quickly. Yes, ammonia is very much toxic to brain, surgery. The ammonia is very much toxic to brain. Okay, so it is toxic to brain, leading to something called as encephalopathy, called as hepatic encephalopathy. Hepatic encephalopathy. What is the drug of choice for hepatic encephalopathy? What is the drug of choice for hepatic encephalopathy? Tell me, come on quickly. Hepatic encephalopathy's drug of choice is lactulose, sir. What is that? Lactulose. So this acute liver failure can be seen with which diseases? It can be seen with the diseases like Ray's syndrome. The drug of choice is lactulose. Drug of choice is lactulose. Is the drug of choice for hepatic encephalopathy. Lactulose is it is a drug which is used in the treatment of constipation also. It is a type of laxative agent. It is useful in the treatment of hepatic encephalopathy also. Now they'll ask you one more question. Urea is synthesized by which amino acid? Urea is synthesized by your both of them arginine, one amino acid called as arginine, and most importantly, the urea cycle occurs where? Very good, Gautam. So, what is that next one? That is urea cycle occurs in both cytoplasm and mitochondria. Both cytoplasm and mitochondria. These are the important question which will come from your, what is that, urea cycle. Okay, it occurs in both cytoplasm and mitochondria. So let's go to the next one. That is only aromatic amino acid. That is your phenylalanine. So before going to the phenylalanine is a type of essential amino acid. Which are the amino acids which are essential amino acids? Essential amino acid. So we have TV till 8 p.m. is the mnemonic. Okay, it is not the 8 p.m. liquor brand I'm talking. It is the mnemonic I'm speaking. Okay. Very good. TV till 8 p.m. What is T stands for? T stands for tryptophan. T stands for tryptophan. V for valine. V for valine. Okay. So, what is that? T for threonine. Okay. Threonine. And I for isoleucine. I for isoleucine. Okay. L for leucine. L for lysine. Okay. P for phenylalanine. M for methionine. M for methionine. So these are the things which you need to remember. TV till 8 p.m. Okay, very good. These are the 8 amino acids which are essential. Okay, let's start off with the first amino acid, phenylalanine. What is that? Phenylalanine. So remember, my dear students, whatever the phenylalanine comes into the body, this phenylalanine undergoes one reaction called as, what is that? Hydroxylation reaction. Which reaction? Hydroxylation reaction. So this hydroxylation is done by phenylalanine hydroxylase. What is the name of the enzyme? Phenylalanine hydroxylase. So this will give rise to your amino acid called as tyrosine. So since tyrosine is formed from the phenylalanine, so we don't need tyrosine to be taken from the diet, because phenylalanine will synthesize the tyrosine. So this OH group is donated by whom? The OH group for the conversion of phenylalanine to tyrosine is done by whom? Done by whom? Everybody should tell me. Very good, Akash Gupta. That is your tetrahydrobiopterin. Tetrahydrobiopterin. Who will donate the OH group? That is donated by your tetrahydrobiopterin. Okay. Now this tyrosine, whatever is there, it forms your catecholamines. It will form your T3 and T4, and it will also form other substances such as melanin, not melatonin, melanin. This is a very important point in all the batches, in all the centers when I'm discussing. I told this so many times, this will confuse you. So yes, char bottle vodka story, remember? All right. So tetrahydrobiopterin. So that will give, what is that? That will give OH group to the tyrosine. So what are the substances which are formed from the tyrosine? Catecholamines. What is that? Catecholamines. Don't think as late. Okay. It is catecholamines. Okay. Catecholamines, T3, T4, and melanin are produced from your tyrosine. Okay. So, so, so melanin is the skin pigment. Melanin is the pigment which is present in your skin and as well as in your hair. If there is a deficiency of phenylalanine hydroxylase, no tyrosine. If there is no tyrosine, catecholamines are not there. T3, T4 is not produced. Melanin is reduced. So if melanin is reduced, skin becomes pale. So skin becomes pale, hair becomes blonde. So it is like, white skin, blonde hair, very beautiful appearance. But the problem rises with what? T3, T4 also not there. So here what will be there? Person looks beautiful, but brain development will not be there. Why? Because T3, T4 is absent. So because of that, brain development will not be there. Now more phenylalanine will be present in the body. This will lead to a disease called as phenylketonuria. What is the disease called as? Phenylketonuria. Okay. So if there is a deficiency of phenylalanine hydroxylase, there is no formation of tyrosine, leading to phenylketonuria caused by the defect called as phenylalanine hydroxylase gene, PAH gene mutation. Okay, so because of that, what are the clinical feature? Pale skin, blonde hair, very beautiful, but don't fall for that. Mental retardation will be there, and also mousy odor are possible. Because why mental retardation? For the brain development, we require T3 and T4. If T3 and T4 is not there, brain development will not take place, leading to phenylketonuria. Any confusion so far about phenylketonuria? Any confusion? All right. So what is this? That is your phenylketonuria. This is a very commonly asked question. So don't fall for the beauty, because there can be possibly, what is that? Possibly this problem, mental retardation. Let's go to the next one. That is only where, yes, your tyrosine produces T3 and T4. So let's understand the synthesis of T3 and T4. Okay, let's understand the synthesis of T3 and T4. Okay, so first what happens, my dear students, whenever you eat the food, which consists of iodine, okay. So to understand iodine will be absorbed by your thyroid gland. Whenever your body has iodine, all the iodine will be absorbed by your, what is that, absorbed by your, yes, thyroid gland. So once thyroid gland absorbs the iodine, so in the thyroid gland, we are always, we remember, so if you consider this is a thyroid gland, thyroid gland has a follicle called as thyroid follicles. If I take one follicle and put it outside, so this follicle will have a epithelium called as follicular epithelium. Inside the thyroid gland, whatever is there, called as colloid. Okay. So it is like many students will get bored by listening to this, especially colloid, because where and all you studied thyroid gland? Tell me, related to colloid, in which the, how many subjects you studied? You studied it in biochem. So many are there. Biochem. Okay, you studied it in pharmac. You'll study it in medicine also. Anyways, colloid is there, sir. Okay. Now colloid is there. So I will take one epithelium and put it here. So this is one epithelium. So this is the other side of the epithelium. So in this, we will have something called NIS, sodium iodine importer. Sodium iodine importer. It takes up your, what is that? It takes up the sodium and also your iodide. It takes up the sodium and iodide. Sodium and iodide comes into the cell and it will undergo the formation into iodine. It will undergo the formation into, what is that? Iodine. Okay, it will formation of iodine. Okay, now, whenever your stimulation of TSH receptor, whenever there is a stimulation of TSH receptor, they synthesized a protein called as thyroid globin. Protein called as thyroid globin. Thyroid globin is represented as TG. TG also comes into the colloid. Don't get confused. So for iodine came into the colloid, TG came into the colloid. Okay. So for this iodine to enter inside the cell, we have special pump here, transporter here called as pendrin. Okay. So pendrin is the apical iodine transporter, which is responsible for what? Which is responsible for transport of iodine from the follicular epithelium to the colloid. So inside the colloid, iodine and thyroid globin came. So thyroid globin is a protein. All the protein consists of made out of amino acid. In the thyroid globin, we will have an amino acid tyrosine. Okay, to this tyrosine, I will add up the iodine. I will call it as a iodination and coupling. Because of that, for the coupling, we will require some enzyme for iodination and coupling. We will call it as a TPO. That is only called as thyroid peroxidase. So thyroid peroxidase will synthesize your monoiodotyrosine, diiodotyrosine, T3 and T4. T3 and T4. Now, whenever your body requires T3 and T4, that colloid droplets will come. That droplet will be secreted into the blood. That is only your conversion. That is only your formation of T3 and T4. Formation of T3 and T4. Clear, everybody? T3 and T4. Okay. So which amino acid is playing a role? Tyrosine. If everybody understood this, if you tell me yes, I will add some important points to this thing. Understood or not? Okay, all right. अगर समझ गए तो आगे बढ़ेंगे चलिए let's look at the understanding. So first few important points which I would like you people to know, my dear students, is that is your TPO inhibitors. TPO inhibitors. TPO inhibitors are used for which disease? Used for hyperthyroidism. Used for hyperthyroidism. Okay. So which are the drugs we have? We have carbimazole, methimazole, and PTU. We have carbimazole, we have methimazole, and PTU, which is the safe in first term pregnancy, safe in first trimester pregnancy, first trimester. Okay. So first trimester safe. These two are used for second and third trimester, because propyl thiouracil cannot be used for a longer period of time. If you use it for a longer period of time, what will happen? Yes, that will lead to a problem called as fulminant hepatitis and agranulocytosis, but it does not affect the fetus. That's why we are using it in the first trimester. TPO is used for the hyperthyroidism. Then the next one, my dear students, in the blood itself. So what happens to the quantity of T3 versus T4? Understand quantity, first quantity. Okay. With respect to quantity, yes, your T4 is more quantity than T3. Okay. And one more point I want you people to remember is, yes, apart from the quantity, potency. Potency means ability of its action. T4 is more T3 is more potent than T4. T3 is more potent than T4. T4 is less potent. T4 is less potent. So in the blood, whenever your body requires more amount of T3 suddenly, let's imagine you suddenly go to Russia now. Russia, for example, I'm taking it as an example of like, for this thing, okay? Cold country. Okay? Guys, somebody's phone is unmute and you are going near to some TV. I think it is creating noise. Please look into that. Now, anyways, we have this one. What is that? So suddenly went to Russia. Your body has to synthesize new thyroid, means your body requires some time. So till then, your body should be able to manage. So in your body, what your body will do, it will convert in the blood, it will convert T4 into T3. This conversion occurs with the help of an enzyme called as deiodinase. Deiodinase. This was a previously asked question. Peripheral conversion of T4 to T3 takes place by which enzyme? That conversion will take place by deiodinase, removal of iodine. So this question was previously asked. So please don't get confused. Clear, everybody? Clear? Now, melanin. Next one is melanin. Melanin is produced in melanocytes, and melanocytes are derived from neural crust cells. Melanocytes embryologically derived from neural crust cell. So, neural crust cells, my dear students, forms what and all it will form. Let's understand what are the derivative of NCC, neural crust cell derivatives. Okay. So, derivatives of neural crust cells. Okay. Let's see. So, one is your melanocytes, which already I have told you people, melanocyte. Then in the thyroid gland, we have some cells called as C cells, called as C cells. Okay, C cells, which produces what is that? Which produces calcitonin, and melanocyte and your adrenal medulla. All three of them are derived from your neural crust cell. Okay. So now what will be there? We have one tyrosine. We have an amino acid called as tyrosine. Tyrosine is getting converted into melanin. Tyrosine is getting converted into melanin with the help of what? With the help of, very good. The answer is adrenal medulla is a correct answer. Now let's understand. Tyrosine to melanin conversion takes place by the enzyme. What is the name of the enzyme called as? Written there, only read it. What is the name of the enzyme? Very good. Repeat, everybody. Tyrosinase. Repeat, what is the name of the enzyme? Tyrosinase. Tyrosine will convert into melanin with the help of an enzyme called as tyrosinase. Okay. So, if there is a deficiency of tyrosinase, so throughout the body that tyrosinase is not there. So throughout the body, what is not formed? Melanin is not formed. Throughout the body, melanin is not formed. So all over the body, if melanin is absent, we will call this disease as albinism, called as albinism. But what is that? Albinism. So there is albinism. So it is full body depigmentation or only patchy depigmentation? It is a full body depigmentation. Full body depigmentation. Very good. That is full body. Very good. Correct answer. Correct answer. Both of you. Okay. Depigmentation. Then vitiligo. Vitiligo is a disease in which what happens is that patchy depigmentation. Patchy depigmentation. Okay, so patchy depigmentation, so white patches are formed. Okay, so that occurs as a result of failure of migration and autoimmune destruction of the melanocytes, leading to what is that? Vitiligo. So vitiligo is a patchy depigmentation versus albinism is a full body depigmentation. Okay, so hope that is clear for you people. I'll continue to the next aspect, synthesis of catecholamines. Okay, synthesis of catecholamines. Okay, so I will try to discuss catecholamines. What is that? Synthesis and destruction, both of them together. Okay, so we have an amino acid called as phenylalanine. Okay, that went into conversion of tyrosine. Now tyrosine itself is an individual amino acid. So let's start off with the tyrosine itself. So tyrosine, my dear, undergoes hydroxylation, OH, adding of the OH group. So add the tyrosine to the OH, we will have, with the help of an enzyme called as tyrosine hydroxylase, we get DOPA, dihydroxyphenylalanine called as DOPA. Okay, that dopa, my dear students, undergoes one reaction called as removal of carbon dioxide. Removal of carbon dioxide with the help of an enzyme called as dopa decarboxylase. Which enzyme? Dopa decarboxylase. It will give rise to dopamine. It will give rise to dopamine. Now, this dopamine, my dear students, undergoes hydroxylation once again. So, if you add OH group to the dopamine, we will get noradrenaline. Okay, noradrenaline. So this noradrenaline undergoes one reaction called as methylation. What is methylation? Adding a methyl group. What is methyl group? What is methyl group? Wolff-Chaikoff phenomena. For sure, I will explain. No problem. Not a problem. I will explain. Let me finish this one and I'll go back to Wolff-Chaikoff. Very good. That is your CH3. Okay, methyl group means CH3. You are adding a which group? Methyl group. That is your addition of CH3 group. CH3 group is being added. If you add the CH3 group, it will give rise to adrenaline. It will give rise to adrenaline. Okay, so this is the synthesis of catecholamine. Okay, now these catecholamines can undergo what is that? Breakdown also. Catecholamines aayega, they will act on the sympathetic nervous system and they undergo the breakdown. So dopamine, my dear students, it will break down into something called as homovanillic acid. Homovanillic acid. Okay, homovanillic acid, and noradrenaline will get converted into normetanephrine. Adrenaline will get converted into metanephrine. Means they're formed. They do their function. After doing their function, what will happen, sir? After doing their function, what will happen? They undergo the breakdown. So once they break down, it will give rise to, one is metanephrine and one more is called as mandelic acid, or also called as VMA. Okay, HMMA or VMA, vinyl mandelic acid. Okay, so simple as that. The breakdown products are your metanephrine. Nephrines are the breakdown product. Metanephrines are increased in which disease? See, more adrenaline, more metanephrine. More adrenaline, more metanephrine. In which disease metanephrines will be elevated? Everybody should be able to tell what is the disease. Very good, very good. What is that? So Abhishek and Manivel, both of you are correct. What is that? That is increased in pheochromocytoma. Pheochromocytoma. Quickly, let me give you the gist of the pheochromocytoma. Okay. By the way, since I told gist, what is GIST? Capital GIST. What is the full form of GIST? Not GST, GIST. Gist. What is this full form? What does gist means? Yes, it is a type of gastrointestinal stromal tumor. Gist is a gastrointestinal stromal tumor. Very good. Okay. Anyways, let's quickly discuss about pheochromocytoma. Pheochromocytoma, everything is written in the notes. You would have read it in your medicine already, but I'm going to recap that once again for you people. Just listen to me. Pheochromocytoma is a type of adenoma. Pheochromocytoma is a type of what? Adenoma. Adenoma means it is a type of secretory tumor, means it secretes something. What it will secrete is metanephrines. Oh, sorry, it will secrete is adrenaline, excessive amount of adrenaline. So that is the definition. Next one is what? There is an excessive amount of adrenaline production. So if more amount of adrenaline production, excessive activation of sympathetic nervous system. So excessive activation of sympathetic nervous system leading to clinical features such as hypertension, blood pressure can elevate. Okay, patient will have hyperglycemia because sympathetic activation increases the glucose production. Okay, then also patient will have an occipital headache. Okay, so there might be, whenever there is a headache, the patient might have a vision disturbance, but not mandatorily. And all these symptoms are episodic, means when it secretes more adrenaline, that time symptoms will come, later it will disappear. Okay, so during the episodes of elevated blood pressure and occipital headache, there is a hyperglycemia during that time. Then You suspect a pheochromocytoma, how do you look for? So we need to know the location of pheochromocytoma. Pheochromocytoma is such a type of tumor which is most commonly originates in the adrenal medulla, but also it can originate from other places. So it is both adrenal and extra-adrenal tumor. Adrenal and extra-adrenal tumor. So for the investigation, so we use 24-hour urine metanephrine level as a measurement. 24-hour urine metanephrine as a measurement. 24-hour urine metanephrine. So if 24-hour urine metanephrines are elevated, that is suggestive of pheochromocytoma. We can do a nuclear scan called as MIBG scan, or we can go for CT or MRI abdomen for the finding the location. But best investigation nuclear scan we use is MIBG scan. Which scan? MIBG scan. Which scan? MIBG scan. Okay, MIBG scan is used, and after you find the tumor, so first because patient will have elevated blood pressure, you need to control the blood pressure. To control the blood pressure, we give a drug that is called as phenoxybenzamine. Repeat everybody. Phenoxybenzamine. Phenoxybenzamine is a irreversible alpha blocker. But phenoxybenzamine can also be combined with one more drug. What is that? Can anybody tell me what is the combination what we will combine with phenoxybenzamine? MIBG is the best, sir. MRI or MIBG matlab MRI. Okay. Yes, we need to combine propranolol. Very good. Very good. All of you are correct. Beta blocker, that is your propranolol, should be combined. Okay. So after you combine this, after you combine the propranolol, we can control the blood pressure. Later, what we have to do? Later, we will surgically remove the tumor. This is the story of pheochromocytoma. So there is always some questions which have been asked in the paper, one or two related to pheochromocytoma. So I don't want you people to make any mistake. That's why I discussed the whole thing, though it is not a syllabus of biochemistry. Irrespective of that, I'm trying to make sure that what is that you understood the pheochromocytoma. Okay, let's go to the next one. Degradation of tyrosine. What is that? Degradation of tyrosine means how does tyrosine breakdown occurs? Whatever tyrosine is there, tyrosine did its activity, now it has to break down. So breakdown of tyrosine or degradation of tyrosine and phenylalanine. So phenylalanine get converted into tyrosine. Now this tyrosine, under the influence of an enzyme called as tyrosine aminotransferase, it forms something called as 4-hydroxyphenylpyruvate. 4-hydroxyphenylpyruvate. 4-hydroxyphenylpyruvate will give rise to what is that? Homogentisate. Homogentisate. Now this homogentisate, under the influence of, remember, homogentisate oxidase. Homogentisate oxidase, it will give rise to mallyl acetoacetate. That mallyl acetoacetate will give rise to fumaryl acetate. Fumaryl acetate. This fumaryl acetate, under the influence of fumaryl acetoacetate hydrogenase will be there. So that will give rise to, that will break into what is that? Fumarate and acetoacetate. Sir, you told in the classroom also this pathway is not important. So what exactly we need to remember? Okay, remember there is a disease called as which disease? Alkaptonuria. What is that? Alkaptonuria. What is that? Alkaptonuria. Alkaptonuria is a condition. What will happen, my dear students, is there is a deficiency of which enzyme? Can anybody tell me deficiency of which enzyme alkaptonuria is due to? Come on, guys, quickly. Everybody should tell me. Come on. Cheppandi. Solunga. Bolo. Very good. Alkaptonuria is due to deficiency of which enzyme? Homogentisate oxidase. Homogentisate oxidase deficiency. Homogentisate oxidase deficiency. If there is a homogentisate oxidase deficiency, there is an increase of homogentisate that will enter into urine. So once it enter into the urine, it undergoes oxidation when exposed to air. So what will happen to the urine? It will create a black color of the urine. Upon air exposure. So normally when the person urinates, urine color will be normal, but upon exposure to air, the urine turns into black color. We'll call it as a alkaptonuria. And also this homogentisate can deposit in the form of benzoquinone acetic acid in the tissues. It deposits in the multiple parts of the body, which will lead to ochronosis, pigmentations in the eye. Pigmentation in the eye, it can get deposits in the it can get deposited in the tendons, leading to musculoskeletal defect. So these are the symptoms which are associated with your alkaptonuria. So alkaptonuria is a deficiency of which enzyme? Deficiency of your, yes, that is only called as, that is only homogentisate oxidase. So here I want you to remember one more thing here. So there is one commonly encountered disease which I would like you to remember that is only called as hereditary tyrosinemia. Hereditary tyrosinemia 1. Hereditary tyrosinemia type 1. So this is not told in any batches. In any batches, we haven't discussed this. So please write it down. Hereditary tyrosinemia are also called as tyrosinemia type 1. Tyrosinemia type 1. Tyrosinemia type 1. It occurs due to deficiency of which enzyme? Deficiency of your, that is your FAH. What is that FAH enzyme? This is your FAH. That is your fumaraldehyde, fumaryl acetate hydrogenase. Fumaryl acetate hydrogenase deficiency leads to what is that? Type 1 type 1 tyrosinemia type 1. So that is a very important question to be remembered. Okay. Let's go to the next important aspect. So with that much being said, we are done with what is that important aspects of amino acids. Now that is not done yet. We need to discuss about the metabolism of one more important that there are important amino acid. So first let's start off with your glycine, sir. What is that? Glycine. Glycine is a amino acid. It is a simplest amino acid. FH full form. Fumaryl acetoacetate hydrogenase. This is the short form called as FH. Understood? Understood, Hemant? FH? Okay. Glycine is an amino acid which is the simplest amino acid. So my dear student, the simplest amino acid glycine, whatever is there, what are the functions of glycine? Glycine is responsible for heme synthesis. Glycine is responsible for heme synthesis. It is responsible for creatine synthesis. And there is one more tripeptide called as glutathione. Okay, glutathione. So one more tripeptide is called as glutathione. Then it is responsible for the purine ring synthesis. It is present in one of the amino acid, abundantly present. Abundant in collagen. Collagen is a protein, sir. In this protein, what is abundant? Most abundant protein means when you take collagen, let's imagine for a number, you have 1,000 amino acids. Among those 1,000, 200 amino acids will be glycine only. Rest everything will be combined with the other 800. So which one is most highest in quantity will be glycine. In where? In the collagen, and also it is responsible for the conjugation of the bile. So these are the functions of your glycine. Once we understood about the functions of glycine, let me recall and recap about one of the most important amino acid, that is your sulfur containing amino acid metabolism. So sulfur containing amino acid metabolism, I want you to understand very simple aspect. So this one, many students had a confusion during the class also. They used to ask me to repeat. So now that time also you have read it, sir, but I'm forgetting. So that for that purpose only I'm explaining the whole thing once again, because the two major chapters for your biochemistry is your amino acid and carbohydrate only. That's why I'm going very slowly. Okay, hope this is getting very useful for you people, I hope, sir. We have your noradrenaline. Noradrenaline undergoes conversion into adrenaline. So what is the reaction which is occurring? That is your methylation. Okay, methylation. Okay, so we have one amino acid, that is your cysteine. What is that? Cysteine. Okay, cysteine donates. Cysteine is present in the form called as SAM, S adenosyl homocysteine. It is present in a form called as S-adenosyl homocysteine. S-adenosyl methionine. Sorry, why am I making mistake? Sorry, S-adenosyl methionine. It is present called as methionine. S-adenosyl methionine called as SAM will be present. Okay. Now this SAM, whatever is there, sir, this SAM fellow will donate your what is that methyl group. Donates the methyl group to what? Methyl group to noradrenaline, and it will form the adrenaline. After removal of the methyl group, it will form adenosyl homocysteine. Okay. All right. So S-adenosyl homocysteine. नहीं कभी कभी हो जाता है. Okay, कभी कभी हो जाता है. So we have what is that? SAM, S-adenosyl methionine is there. Methionine donates the methyl group. S-adenosyl methionine donates the methyl group to noradrenaline. So noradrenaline becomes adrenaline. Okay, clear everybody? कोई confusion नहीं है ना अभी? जमील, hope that is clear now. अभी कोई confusion तो नहीं है ना? चलो. Now, what is the difference between abhi homocysteine hai, methionine hai. We have homocysteine and methionine. Here is our homocysteine, here is our methionine. So, after C, simple as that, whenever methionine is there, methionine minus one methyl group is equal to homocysteine. That's it. Is equal to that of homocysteine. Okay, don't get confused. Methionine, once if you remove a methyl group, homocysteine. Okay, now if you have a homocysteine, so to the homocysteine, if you add one methyl group, that is equal to methionine. So what is the difference between any form of methionine and any form of homocysteine? Any form of methionine and homocysteine, difference itna hi hota hai. Difference is this much only. What is that? Difference is, my dear students, CH3 group. So now this homocysteine, whatever is there, it will get adenosyl homocysteine will get converted into homocysteine. Okay, now this homocysteine will form methionine. So if I want to form methionine from the homocysteine, what group I need to add? What is required for addition to homocysteine? What I have to add so that it becomes methionine? Tell me. Look at this one and tell me. What I have to add into homocysteine so that it becomes methionine. होमोसिस्टीन के अंदर मैं ऐसा चीज, कौन सी चीज ऐड करना चाहिए ताकि वो मेथियोनिन बन सके? That will be your CH3. So I need to add a CH3 group. That CH3 group, my dear students, this comes from our food called as folic acid. This thing called as folic acid, B9. Okay, now from the folic acid, I'm taking the methyl group and adding it into homocysteine. So I require an enzyme called as that is homocysteine methyl transferase. Homocysteine methyl transferase. So homocysteine methyl transferase collects the methyl group from where? From the folic acid and adds it to the homocysteine so that it will become into methionine. Okay, so this methionine, whatever is there, this methionine will get further converted back into SAM, and this cycle keeps on repeating. Okay, sir, this is what is the metabolism of your sulfur containing amino acid methionine. Okay, so now if there is a folic acid is not there, automatically homocysteine, will it get converted into methionine? No. So homocysteine level will rise. If folic acid is not there, no methyl group. If no methyl group, homocysteine will not get converted into methionine. So deficiency of folic acid will lead to increased level of what is that? Homocysteine. Okay, vitamin benign deficiency. Benign deficiency will lead to increase of homocysteine. Increase of homocysteine. Remember homocysteine. Why increase of homocysteine? Increase of homocysteine because of one reason, because there is no methyl group for its conversion into methionine. Okay. Now let's look at this homocysteine methyl transferase. It requires vitamin B12. Okay, the enzyme requires a coenzyme called as vitamin B12. So this coenzyme vitamin B12, whatever is there, coenzyme vitamin B12 helps the function of homocysteine methyl transferase. So if B12 is not there, this enzyme will not work. If enzyme will not work, automatically homocysteine level will be raised. So in the B12 deficiency also, we will have a elevated dry level of increase of homocysteine. So both of them causes megaloblastic anemia. Both of them causes increase of homocysteine. So both of these deficiencies, common symptom is megaloblastic anemia. Common symptom is megaloblastic anemia. Okay sir, then biochemically इसको differentiate कैसे करें sir? How do we differentiate B12 deficiency with the B9 deficiency if they give only about homocysteine? So is there any other parameter, biochemical parameter? Of course, in this remember there will also be elevated level of MMA. What is that? Elevated level of MMA, that is methyl malanoic acid. So this is the other parameter which we check. So both of them are elevated, B12 deficiency. If only one is elevated, B9 deficiency, that is homocysteine. Do not get confused. Clear? So this is a story of your sulfur containing amino acid. So megaloblastic anemia will be there. So quickly let me tell you, B9 deficiency is a folic acid. Folic acid deficiency leads to megaloblastic anemia. B12 deficiency also leads to megaloblastic anemia. That is in which what will be elevated in both of them, homocysteine level is elevated. Homocysteine level is elevated, but in B12 deficiency MMA level is elevated. इसको star लगा दो भाई साहब, बहुत important point. What is there? That is your megaloblastic anemia and homocysteine related. Okay. All right. I'm hoping that सबको ये समझ आ गया, अंदर के अर्थమైందా, पुरुंजिदा पनियातना समझ में आया? सल्फर कंटेनिंग अमीनो एसिड्स आया ना? ये तो बताओ भैया. Понятно? Хорошо. Хорошо. All right. Let's continue. Let's continue what exactly we need to discuss further. So in the amino acids chapter, I have discussed all the important discussion which has to be done. Okay. So the next important thing which we need to understand, my dear students, is your histamine. Okay. What is that? Histamine. Now histamine comes from an amino acid called as histidine. Okay. अगर हमारा नसीब खराब होगा ना, कभी पूछ भी लेता है, histidine undergoes which reaction? Histidine undergoes decarboxylation reaction called as decarboxylation reaction. Decarboxylation reaction to form histamine. To form what is that? Histamine. Now histamine will be freely present or it will be stored. It will be stored whenever our body requires histamine. It will release. In the brain also it is present. It is present in the stomach. It is also present in the mast cells. So histamine is produced majoritarily stored in the mast cell. This is a पूछा हुआ question है, ठीक है? वहां जाके फिर ऐ करके मत करना. All of you remember they'll ask this kind of lame questions also sometimes. Histamine is produced by which cell? Produced by mast cell. Okay sir, इतना simple question आता है कि एग्जाम में? I have told you people, every batch, every student, I have told you people, and I mean every place, be it Chennai, be it Trivandrum, be it Delhi, or in every place I have told, not all the 300 questions are complex. Not all the 300 questions are confusing. There will be 60 to 70 questions which are simple baseline logic ones, basic fundamental things. After that 70-80 questions only, slowly the difficulty level will start to rise. I don't want you people to lose out any time also in the confusion, and also I don't want you people to lose out by lose out anything by getting confused. And by I don't want you to lose out these questions which are very easy. ये दोनों पे नहीं होना चाहिए आप लोगों को. When you have a simple question, when you know the answer, mark the answer, leave it at that point, get ahead with the next questions. Okay, ऐसा चीजों में बहुत बहुत fast रहना चाहिए आप लोग. Okay, anyways, so we are done with what is that histamine. So with that much being said, we are done with what is that amino acid. Let's go to the next one called as proteins. What is that? Proteins. Now when we are speaking of proteins, based on their structure, we have primary. Primary means a simple one line of amino acid, one chain of amino acid is there, we'll call it as a primary proteins. Then we have little bit complex called as secondary. That means simple structure, but it will have a two lines, maybe two lines are present, means two chains of amino acid can be present, secondary. Then we have tertiary. So what is this tertiary proteins? Tertiary proteins will have some twisted or something like that. And the last but not the least, the most complex protein is your quaternary protein. Okay, so quaternary is the most complex protein which will undergo folding and it will have a three-dimensional shapes. Okay, so the next important quaternary proteins. So quaternary proteins have something called as protein folding. Protein folding is done by whom? Protein folding is done by whom? Come on, quickly. Skajit Pajalusta, tell me please. Protein folding is done by Chapri. That is your chaperones. What is that? Chaperones. Chaperones are a support protein or a auxiliary protein. Very good, all of you are correct. So support protein or a auxiliary protein which is responsible for the protein folding. Okay, then protein death is caused by which substances? Proteins are killed by what? Proteins are killed by, all of you know that, tell me. That is one last kiss of the protein, that is kiss of death. Kiss of death is which one? Ubiquitin. Very good, but what is that? Ubiquitin. Very good answer, everybody is correct. Ubiquitin. What is that? Ubiquitin. Ubiquinone is coming where? Ubiquinone is coming in electron transport chain. Okay, so now we have some other way we can classify. Some of them are called as plasma proteins. So these and all you people will know. So still I'm going to quickly recall about that. Okay, plasma protein, most abundant one is which one, sir? Most abundant plasma protein is albumin. Most abundant plasma protein is albumin. Then we have globulin. Okay, and your fibrinogen. All of them are your, what is that? All of them are your plasma proteins. So albumin, okay, globulin, fibrinogen, all of them are your plasma protein. There are a lot of them are ceruloplasmin. Okay, there we have, for example, protein C, protein A. So any protein which is present in the blood plasma called as plasma protein, among which एक को तो याद करिएगा sir, albumin. Albumin maintains, what is the function? What it will maintain? Maintain oncotic pressure of the blood. Oncotic pressure of the blood. It maintains the oncotic pressure of the blood. So please understand, that is your blood's oncotic pressure is maintained by your, what is that? That is your albumin. Let's go to the next one. That is your collagen. Okay. Now collagen के बारे में थोड़ा पढ़ा देता हूँ. What is this? Listen to me. Now anytime, for example, when we are looking at collagen, so first we have DNA. From the DNA, we will form mRNA. What do you call this process as? DNA to mRNA, what do you call it as? DNA to mRNA, क्या बोलते हैं उसको? DNA to mRNA, we will call it as transcription. Okay, transcription. Okay, transcription. This transcription will go and form the, so transcription from mRNA to protein, we will call it as translation. Followed by the translation, sir. We will have, what is that? We will have Post translational modification. Post translational modification means a protein is formed. After forming the protein, there will be changes. What are the changes? Maybe we add some group to some amino acid, or maybe we fold the protein. Okay, we change some, alter some structure in the protein. All the changes which occurs after the translation, we call them as a post translational modification. What do we call it as? A post translational modification. What is it called as? Post-translational modification. So what is a post-translational modification? Changes which occurs after the translation. So post-translation modification, collagen undergoes post-translational modification. What is the post-translational modification in collagen? I taught you people. Post-translational modification in collagen. What is that modification? What is the change which is occurring? Which is occurring? So in the collagen, we will have proline, sir. We will have proline. We will have an amino acid called as proline. That proline will get converted into hydroxyproline. Hydroxyproline. Proline will get converted into hydroxyproline. So this is the post-translational modification which occurs with, what is that? Occurs with the collagen. So for that post-translational modification, for that hydroxylation, we will require one vitamin called as vitamin C, sir. We require one vitamin called as vitamin C. Okay. Vitamin C. Okay. All right. Let's continue to the next one. What is that? So that is your post-translational modification requires vitamin C. That what is the modification? Modification is your conversion of proline into hydroxyproline. Now hydroxyproline, is it genetically coded? It is formed after the translation. It is not genetically coded. It formed after the translation. That is why we call it as a derived amino acid. So that is why hydroxyproline is called as derived amino acid. Means the derived amino acid is not coded in the protein, rather it will be formed after the formation of the protein. We will call it as a hydroxyproline. So that is the story of your, what is that? That is the story of your, yes, post-translational modification of the collagen. So collagen is divided into multiple types. We have type 1 collagen. So repeat with me. Type 1 collagen is located in ligaments. Where is it located? Ligaments. Type 1 location is located in bone. Repeat with me. It is located in the bone. Dermis and tendon. Dermis and tendon. If there is a defect, it will lead to a disease called as type 1 collagen defect. We will call it as a osteogenesis imperfecta. Osteogenesis imperfecta. Osteogenesis imperfecta. Can you tell me some symptoms from your osteogenesis imperfecta from your orthopedics, bacho? Bone defect will occur. So can you tell me some important clinical features? So there is one blue color sclera, easy fracture. There is a developmental defect of the bone. Okay. So blue sclera. Okay. Then there is a deformity. What is that deformity? CT. What is that? CTEP deformity. Okay. Okay. That is your CTEP deformity. Okay. So what is that? Congenital talipes equinus varus. Congenital talipes equinus varus deformity. Type 2 collagen is located in the cartilage. If there is a defect, it will lead to a disorder called as chondral dysplasia. Type 3 collagen is located in the aorta and as well as in the skin, sir. Aorta and skin, both of the places it is located. So if there is a type 3 collagen defect, the patient will develop something called as Ehlers-Danlos syndrome. Ehlers-Danlos syndrome, what will happen to the person? There is an excessive stretchability of the skin. Along with the excessive stretchability of the skin, patient will also have risk of aortic dissection. There is aortic dissection, and also they will have a risk of aortic aneurysm. So aortic dissection and aneurysm both are possible with the Ehlers-Danlos syndrome. Then we have a type 4 collagen defect. This is a very important disease. So type 4 collagen is located where? In any basement membrane. It is located in the basement membrane. So automatically, if basement membrane is defective, it will lead to defect of the capillaries and your defect of the glomerulus. Defect of the capillaries and defect of the glomerulus. So that is you will lead to a disease called as, yes, Alport syndrome. It leads to a disease called as Alport syndrome. In a patient with Alport syndrome, it is a type of X-linked recessive disorder. X-linked recessive disorder. What is that? X-linked recessive disorder. Okay, so the patient will have anterior lenticonus, so excessive curvature of the lens towards the anterior side, sensory neuronal hearing loss, and patient will have glomerulonephritis because glomerulus also has basement membrane. And if you take a biopsy, you can find basket wave appearance. What is that? Basket wave appearance. Okay, so let me show you people anterior lenticonus. Anterior lenticonus. Let me find the picture. Where did it go? So this is your anterior lenticonus. This is your anterior lenticonus because why, sir? We know ophthalmology. Don't worry about it. So there are two terms which will confuse you people. Bacho, one is lenticonus. One more which will confuse you, keratoconus. Okay, what is keratoconus versus what is lenticonus? I don't want the people to be confused. So let me explain the difference between both of them. So keratoconus versus lenticonus. Keratoconus is nothing but it is a excessive curvature of the cornea. Excessive curvature of the cornea. So cornea excessive curvature, we will call it as what is that? What is that we will call? Yes, keratoconus. If lens curvature is more, we will call it as a lenticonus. Okay. So look at these two images, we will understand. So in this image, if you notice, cornea is different, but here if you notice the lens is having a curvature towards the anterior side. We will call it as anterior lenticonus. Okay, so that is your anterior lenticonus. Okay, now if cornea itself is bulging forward, then we will call it as a keratoconus. We will call it as a keratoconus. So don't get confused. Keratoconus is a curvature of bulging of the cornea. Okay, okay, curvature of the, what is that? That is your, what is that? Curvature. So curvature of the cornea, and anterior lenticonus is a curvature of the lens. Sir, Alport's is a X-linked dominant. Predominantly it is X-linked dominant, but there are some cases of X-linked recessive possible. It is both dominant and recessive, but predominantly it is dominant here. Okay, so that is a story of your, what is that? That is a story of your, that is a story of your proteins. That is a story of your proteins and amino acids. We have finished. So the next chapter what we have is lipids. Followed by the lipids, what do we have is, my dear students, yes, followed by the lipids we have enzymes and vitamins which has to be discussed. Okay, so before we go for this ones, what is that? This lipids related chapter, I would like to quickly recall about one thing, that is breakdown of heme breakdown, that is jaundice. Related to jaundice, I would like to quickly discuss with you people, then we will go further. Okay, so I want to teach you people one of the most important common this thing, that is only your jaundice. What is jaundice? Jaundice is nothing but yellowish discoloration of the skin, mucous membranes and sclera. Okay, skin, mucous membrane and sclera. So to understand the jaundice, we need to understand about the metabolism of heme. Metabolism of heme. Okay, now whatever heme is there, heme will, so RBC will break down, RBC will die, RBC will die and release what is that? Heme. Now heme is released where? In the spleen. Heme is released where? In the spleen. In the spleen, heme will get converted into something called as a, that is called as unconjugated bilirubin. Of course, before that biliverdin will get converted into unconjugated bilirubin. Unconjugated bilirubin. Okay, this unconjugated bilirubin goes to liver, binds with albumin. Unconjugated bilirubin bind with albumin, bind with the albumin and goes to liver. Goes to liver. Okay, once it goes to liver, but so that in the liver we are talking now. In the liver, what do we have? Unconjugated bilirubin. Unconjugated bilirubin. So this unconjugated bilirubin, whatever is there, it is a type of fat soluble substance. It is a type of which substance? fat soluble substance. So this unconjugated bilirubin will get converted into conjugated bilirubin, conjugated bilirubin. So this conversion takes place with the help of an enzyme called as UDP glucuronyl transferase, Okay. So what is the purpose of conjugation of the bile? So conjugation of the bile purpose is very simple, sir. Conversion of this is a lipid soluble. This will become into water soluble. Okay, water soluble. Okay, so from the liver, conjugated bilirubin enter biliary tree. Enter the biliary tree. Enter the biliary tree along with the bile. So inside the bile, what is present? Conjugated bilirubin is present. So that is enter the biliary tree, goes to intestine, goes to small intestine or duodenum. In the duodenum, conjugated bilirubin can have two possibilities. Conjugated bilirubin will get converted into stercobilin. This stercobilin can go into the stools. That is why stool will always have a yellowish color because of this sterco. Now this stercobilin, instead of going into the stool, it can get absorbed, and it can enter the blood. From the blood, it can go in the urine. When it is going in the urine, we call it as urobilin. What is that? Urobilin. Clear, everybody, so far? Any confusion? Everybody understood? क्या हो गया? किसको वापस जाना है स्कूल? मुझे भी जो हो गया, मैं अभी देख रहा हूँ. Chalo. Anyways, so very good. Once we understood this, let's start off with the first point, sir. In the jaundice, we have hemolytic jaundice. We have something called as hemolytic jaundice. Hemolytic jaundice में क्या होता है? There is a excessive RBC lysis. More amount of unconjugated bilirubin will form. But enzyme, the quantity of UDP glucuronyl transferase is not that much. Means if there is a excessive unconjugated bilirubin forms, this enzyme will become saturated. If enzyme becomes saturated, unconjugated bilirubin cannot get conjugated. Simple as that. Excessive amount of hemolysis, excessive amount of unconjugated bilirubin, but that much amount of enzyme, which is UDP glucuronyl transferase, is not there. Okay, so hemolysis क्या होगा? बढ़ेगा. If hemolysis बढ़ेगा तो क्या होगा सर? hemolysis बढ़ जाएगा तो unconjugated bilirubin बहुत ज्यादा होगा तो ये एंजाइम काम नहीं कर पाएगा, क्योंकि एंजाइम सैचुरेट हो जाएगा. तो उसके वजह से सिर्फ और सिर्फ क्या बनेगा? अनकॉन्जुगेटेड बिलिरुबिन बनेगा. ओके, एवरीबॉडी अंडरस्टूड? नाउ hemolytic jaundice, there is an increase of unconjugated bilirubin. But compared to normal, there is a severe increase of unconjugated bilirubin. There is a slight increase of conjugated bilirubin also. Slight increase of conjugated bilirubin can also occur because body will try to maximize, but it cannot convert the complete thing. And one more thing, what will happen, sir? Yes, increase of hemolysis, unconjugated bilirubin and conjugated increases. So more bilirubin will go into stool, leading to dark stool. Now, more unconjugated bilirubin, this fellow unconjugated bilirubin himself will go in the urine, leading to dark urine, leading to what is that? Dark urine. Okay, that is only hemolytic, are also called as pre-hepatic jaundice. All the hemolytic diseases such as G6PD deficiency, sickle cell anemia, hereditary spherocytosis, all the type of diseases with the hemolysis occurring, all of them will have a pre-hepatic jaundice, in which there is a elevated level of severely elevation is unconjugated, slight elevation is conjugated bilirubin. Okay, if that is clear, I'll go to the next one. The next type of jaundice what we have is called as post-hepatic means the problem is after the liver. are also called as post-hepatic, which is also called as mechanical jaundice. Mechanical jaundice, which is also called as obstructive jaundice. There is a obstructive jaundice. Okay, so in the obstructive jaundice, what happens? Please listen to me. We have biliary tree. So biliary tree में कुछ ना कुछ obstruction होगा. Inside the biliary tree, there are some other obstruction. So because of that, conjugated bilirubin is formed. But conjugated bilirubin cannot go into the intestine because biliary tree is closed. So there is an obstruction in the biliary tree. Obstruction in the biliary tree, obstruction in the biliary tree, obstruction in the biliary tree, that will lead to what is that? That will lead to increase of conjugated bilirubin in the blood vessel, because conjugated bilirubin cannot go into, yes, conjugated bilirubin cannot go into the intestine. So conjugated bilirubin enters into the blood. Conjugated bilirubin, we increase conjugated bilirubin in the blood. So if more conjugated bilirubin, automatically this will go into the urine, creating a dark urine, creating a dark urine. But stercobilin is not there. Stercobilin, if it is not coming in the intestine, feces will become something called as colorless, but we cannot say colorless feces, called as acholic feces we need to tell. Acholic feces will be there. So this is a pattern of obstructive jaundice post-hepatic. Okay. So post-hepatic jaundice causes are cholangitis, cholelithiasis, okay, pancreatic head carcinoma. All right. So let's go to the next one. That is the last but not the least. We have something called as, what is that? Hepatic jaundice. Hepatic jaundice, इसके अंदर कोई fix नहीं है. There is no fixated this thing. So there is no fixated thing that it will happen like this only, it will happen like that only. It is not fixated. It can have both elevated level of what will be the rate limiting step of heme degradation? UDP glucuronate transferase. Throughout that heme degradation pathway, only one enzyme is the rate limiting, that is UDP glucuronate transferase, UDPGT. Okay. So let's understand what is that hepatic. Hepatic under there can be increase of conjugated bilirubin. So conjugated bilirubin elevated condition. What are the conditions with the elevated level of conjugated bilirubin? Elevated level of conjugated bilirubin is seen with one of the most important diseases, that is Dubin-Johnson syndrome. What is that? Dubin-Johnson syndrome. Okay. Then one more is there called as Rotar syndrome. What is that? Rotar syndrome. What is that? Rotar syndrome. Dubin-Johnson and Rotar are the two types of diseases in which conjugated bilirubin will be elevated, sir. What bilirubin will be elevated? Conjugated bilirubin will be elevated. So we used to remember it with one lame mnemonic called as Dr. Do conjugation. D for Dubin-Johnson, R for Rotar. Both of them conjugated bilirubin is elevated. Then we have increase of unconjugated bilirubin. Increase of unconjugated bilirubin. So unconjugated bilirubin is elevated in Crigler-Najjar syndrome. Crigler-Najjar has two types, type 1 and type 2. Okay. So this is a story of your, what is that? Hepatic jaundice. This is a story of your, what is that? Hepatic jaundice. So hepatic jaundice, I am talking only about few syndromes here. Okay, few syndromes here. That's it. All right. So Stevens-Johnson syndrome is what is the, how do you diagnose? What is Stevens-Johnson syndrome? Anyway, syndrome came, so I'm just discussing about Stevens-Johnson. What is Stevens-Johnson syndrome? SJS, quickly tell me. Let's quickly recap about SJS. Yes, you can even write about Gilbert also. Gilbert also comes under unconjugated bilirubin only. So since I spoke about syndromes, I don't know, my brain is telling SJS again and again. Stevens-Johnson syndrome. What is Stevens-Johnson syndrome? Why it will occur? How it will occur? How do you diagnose? How do you prevent it? Quickly. It is a allergic drug reaction. Allergy to the drugs. So allergy ho gaya to hone do allergy to the drug. So what is the problem here? Allergy to the drugs. Okay. Mainly example is your sulfonamides. Which drugs? Sulfonamides. Mainly which drug? Sulfonamides. Allergy to the sulfonamides mainly. So here there is a epidermal necrosis will be there. Epidermal necrosis will occur. Epidermal necrosis. Again, epidermal necrosis is of multiple types. Epidermal necrosis. When there is a epidermal necrosis, look at the body surface area. Okay, if less than 10% of BSA included, if less than 10% of body surface area included, then we will call it as SJS Stevens-Johnson syndrome. If more than 30% is involved, then what? 30% body surface area in epidermal necrosis, then what do you call the diagnosis as? Anybody? सर अचानक से आप दूसरे सब्जेक्ट में वेरी गुड ऑलरेडी आंसर इज दिस सडनली आई वेंट इनटू डिफरेंट सब्जेक्ट डर्मेटोलॉजी एनीवेज दैट इज कॉल्ड एज 10 टॉक्सिक एपिडर्मल नेक्रोसिस ओके toxic epidermal necrosis ओके TEN तो इन दोनों का बीच में 10 to 30% होगा तो क्या हो बोलते हो इट इज कॉल्ड एज ग्रे एरिया इट्स कॉल्ड एज ग्रे एरिया Okay, so epidermal necrosis there, we cannot say whether it is SJS or toxic epidermal necrosis 10. Okay, so this is the story of your what is that? This is the story of your what is that? Diseases associated with your heme metabolism, mainly jaundice, my dear student. That completes our heme, that completes a quick revision of heme metabolism and also proteins and amino acid is done. Then we are left out with your lipids. Followed by the lipids, we have vitamins and enzymes for the discussion. Okay, so let's take a break now. Let's take a break. After this break, we'll start. Okay. So time is what is that? 6:42. So come back by 17. Okay. Take a small break and we will come back and start off. Okay. Is that okay for everybody? Хорошо? Okay. Perfect. хорошо. Очень хорошо.